Hello, this is Gina Piscatelli with a lecture for students in Anatomy and Physiology 2 at Madison Area Technical College. In this lecture, I will discuss the mechanisms that control blood flow throughout the vessels of the circulatory system. This will incorporate what blood flow is, how do we measure it, what is blood pressure, and a concept called resistance. So to begin, we should define these three terms. The first is blood flow. Blood flow is defined as the volume of blood flowing through a vessel over a certain period of time. It's usually expressed in milliliters per minute. The second and third terms, blood pressure and resistance, are central to understanding the mechanisms that either promote or hinder blood flow. Blood pressure is defined as the force that blood exerts against a vessel wall or a heart chamber. It's expressed in millimeters of mercury. We will see, however, that there is a reciprocal relationship between blood and the vessel wall, in that the vessels can also exert pressure upon the blood. So they're exerting pressure on each other, the blood and the wall. And then finally, resistance is defined as all the forces that oppose blood flow. This term is most often used when referring to the friction that blood encounters from the endothelium of blood vessels. So the terms we just discussed are interrelated. It turns out that the two main mechanisms or factors that affect blood flow are indeed blood pressure and resistance. Let's consider blood pressure first. The pressure of blood upon the wall of a vessel, or the pressure of the vessel upon the blood, isn't exactly what makes blood flow. What makes the blood actually move along the inside of a vessel is that when pressure is exerted on a certain volume of blood at a certain location, that blood has to go somewhere. And it has to go somewhere that's experiencing less pressure. You know this instinctively, like when you squeeze toothpaste out of the tube, you need to have the cap off or open. Or when water flows through a garden hose, there's a source of pressure and then there's an outlet at the end of the hose. So similarly, blood flows from a region where it's experiencing high pressure to a region where it will experience lower pressure. We call this a pressure gradient. So pressure at site A versus pressure at site B. And if you're going from A to B, B should be lower. So what you need to remember is that blood flow occurs due to a pressure gradient. Blood flows from a region of high to low pressure. And this delta sign that's next to the P for pressure, delta means change in, so change in pressure. So for short, that's a pressure gradient. Now let's consider resistance. From its definition, any force that opposes blood flow, you already know that the higher the resistance, the less the blood's going to flow. But we also need to understand what causes more or less resistance, or friction, if you will. And there are three factors that contribute to resistance. And that is increasing vessel length will increase resistance. And this vessel length really just means how far blood is traveling from the heart. Another factor that affects resistance is the diameter of the vessel. If you decrease the diameter of the vessel, then you're going to increase resistance. So the diameter of vessels change as you move away from the heart. If you think of the aorta, at least in the arterial system, if you think of the aorta that has a very large diameter, and the aorta gives off smaller branches, so this diameter is going to decrease with distance from the heart. Another factor that can change vessel diameter is vasomotion, and that refers to vasoconstriction, narrowing the vessel diameter, or vasodilation, which increases the vessel diameter. And then finally, the last factor that will increase friction or resistance is increasing the viscosity or stickiness or thickness of the blood. And this is usually constant in a healthy circulatory system, but with disease, it can change. Okay, so if we think of all these things together, blood flow and what we know about pressure gradients and resistance, we can mathematically represent the relationships, blood flow, blood pressure gradients, and resistance. And we see that blood flow is directly proportional to the blood pressure changes. So the bigger the change, the bigger the blood flow. And then it turns out that blood flow is inversely related to resistance. So if you increase the amount of resistance, you decrease blood flow. Now I'm going to address these factors in a little bit more detail, but slowly. So first, let's talk about pressure gradients in the circulatory system and how that facilitates blood flow. Notice in this diagram that blood in a ventricle experiences a pressure of about 120 millimeters of mercury. This occurs during systole. Okay, not diastole, in systole. Blood is then pushed into arteries, or an artery, where the pressure decreases as you move farther away from the heart. So as the arteries carry blood towards the capillaries and body tissues, down at the bottom of the diagram, the pressure of blood against the vessel wall decreases. So this blood is going to move from an area that has high pressure to an area of low pressure, all the way through the arterial system. By the time the blood reaches the smallest vessels in the body, the capillaries, there's only a pressure of about 15 to 30 degrees or 30 millimeters of mercury upon the vessel walls, upon the capillaries. Now let's look at how the pressure varies as blood comes back to the heart. Notice that blood moves towards the smallest veins, venules. Here are the venules, because the pressure gradient is still dropping. In the capillaries, blood exerts a pressure of 15 to 30, but in the venules, only 12 to 18. So, as it returns to the heart, we still see a drop in pressure. Now, this makes sense if you think about how far blood has traveled away from the ventricle, the main source that exerts a mechanical force onto the blood to get it to move in the first place. But also this has something to do with the nature of veins that we've already talked about, and that is that veins are capacitance vessels. That is, their walls are not quite as stiff as those in the arterial system. Okay, now let's consider resistance. Think of resistance as friction which impedes blood flow. Two features contribute to resistance or friction that are the most relevant. The length of the blood moves, or the vessel length, and the diameter of the blood vessel. First, let's consider length. Resistance increases as length increases because blood flow slows down as it hits against more solid tissue of the blood vessel wall. So in this short vessel, the blood encounters the wall of the vessel for a shorter period of time than it does when it's traveling a further distance. So as the blood keeps moving, resistance builds as the blood encounters friction against the blood vessel wall. So in the longer vessel, we have higher resistance and decreased blood flow. Now, considering how diameter of the blood vessel factors in, we see that the wider the diameter, the less resistance or friction occurs. This is because blood in the center of a wide vessel flows unimpeded by the wall. This is referred to as laminar flow. So the blood in the center of the blood vessel is moving smoothly. In smaller vessels, a lower proportion of the blood, or less of the blood in the vessel, is experiencing laminar flow because there's just less of it in the center. And more of the blood encounters the vessel wall proportionally. So in summary, as length increases, so does resistance. And so you get a decrease in blood flow. And as diameter decreases, resistance increases. You'll get a higher resistance, and therefore you'll get decreased blood flow. Now, even though this is relatively constant in a healthy circulatory system, let's talk about the idea of blood viscosity impacting blood flow due to causing resistance. So viscosity refers to a feature of a fluid. How sticky is it? How thick is it? Like water's not very viscous, right? Water moves pretty freely. And in our blood, our circulatory systems, the viscosity is fairly constant. I can't say it, constant, except in some disorders. So we're going to look at conditions that cause increased blood viscosity, which will increase resistance and decrease flow. One example is dehydration. In dehydration, we'll have a high hematocrit due to the lower amount of plasma. So that means there's more cells per volume of blood, and so it's thicker. The blood is thicker. Another example is polycythemia. And in this case, it's a disorder where we have elevated red blood cells and the blood moves sluggishly. I want to say this is one danger of blood doping, which you may have heard about. In blood doping, athletes receive packed red blood cells so that their blood can carry more oxygen, and this will aid the demand placed on their muscles during whatever activity. However, if blood flow is impeded due to this blood doping, it could cause a pretty serious problem, especially in narrow vessels of the capillaries or the coronary circulation. So it's actually quite dangerous for them to do that. Okay, so far we've discussed blood flow in terms of increasing flow or decreasing flow. And what I've meant by these terms is what promotes blood flow or what impedes blood flow. But it's interesting to also look at the speed of blood flow in different parts of the circulatory system. Notice in this table that as blood moves from the aorta to the capillaries, the speed or velocity of blood flow goes down from 1200 millimeters per second to in the capillaries only 0.4 millimeters per second. Okay. This is due to the fact that resistance is increasing with length of the vessel, or the length the blood traveled, and the vessel diameters narrow over time. But then look at the values for velocity of blood as it travels from the capillaries to the inferior vena cava on its way back to the heart. We see that blood flow gets faster. So in the capillaries, it's really slow. But by the time it reaches the vena cava, one of the inferior vena cava in this case, the speed has increased to 80 millimeters per second, right? And that's due to the fact that there's less resistance in veins. The lowered resistance is due to veins getting bigger and bigger in diameter on the way back to the heart. But as I mentioned previously, it's also due to the nature of the wall of veins. They're not nearly as rigid and they have a high capacity for blood volume. You can think of the speed of blood flowing throughout our circulatory system much like the flow of water from a river into a lake and then out of the lake through streams or some other kind of outlet. So generally speaking, water moving quickly in a river, if it all of a sudden encounters this wide body of water, a lake, then the water will move quite slowly. It'll enter the lake at quite a speed, but then it'll really slow down and spread out. But if the landscape is such that this water is going to flow out as well, then it'll speed back up again. This is what happens in our capillary beds. So the arterial system is on the left here. So blood comes in and it's slowing down. It reaches the capillaries, it slows down, but then as it goes through the capillary bed to reach the smallest little venules, it's going to speed up again. And the advantage to that is that slowing down in the capillaries allows for the exchange of materials between the blood and the fluid in the tissues. So we talk about that in the last lecture of the circulatory system. Okay, so now I have some suggestions about how to organize this information. You need to describe how the velocity of blood flow changes as it moves through the circulatory system and relate resistance and pressure gradients to this. So I tend to make a table for this and I think about, okay, well, we need to know about velocity, we need to know about how the pressure gradients are changing, and we need to know what happens with resistance. So I first start with the arterial system from the aorta to the capillary bed. We know that velocity goes down and that the pressure gradient goes down, which is good. That keeps blood moving. We always want it to be high to low, right? But the reason the velocity goes down, even though there's still a good pressure gradient, is that the resistance has gone up with distance from the heart. Then I take the circulatory system, all the same three factors, and consider blood flow from the capillary bed to the vena cava or back to the heart. We know that it speeds up. And first of all, it's moving at all because there's still a pressure gradient from high to low. But why does it speed up? Because, distance has still increased. It speeds up because resistance goes down due to the fact that the vessel diameter is getting bigger. So that's just my suggestion for answering some study questions that you have. So what we've talked about so far is the velocity of blood flow in the arteries and the veins. Velocity slows in the arteries due to increased resistance. Okay, so I want to discuss some other features of blood flow in the arterial system. Okay, and from the arterial system, then we'll go to capillaries, and then we'll go to veins, and incorporate what you know about the histology between arteries, veins, and capillaries. So first, let's talk about blood flow in the arteries. And notice that blood flow is pulsatile. Okay, what does that mean? That means that it varies greatly. So it peaks with ventricular systole at 120 millimeters of mercury. And yet at diastole, the pressure is 80 right at the top of the aorta. So that's quite a big range. That's a range of 40 millimeters of mercury. So very variable, highly variable, and most variable in the aorta. Now the structure of arterial walls accommodates this pulsation, okay, the pulsatile nature. And blood flow in the arteries is maintained by a pressure gradient, high to low, that we've already talked about, but also due to the elasticity and recoil of arteries due to their tunica media. So we're going to look at the tunica media again after we look at this graph. So this is a graph that shows you the ranges in blood pressure in different vessels throughout the circulatory system. So first we start with the aorta. And the aorta, there's a huge range. But notice that by the time we get down to the very small arterioles, there isn't as much variation. That's because it's farther away from the heart, so the blood isn't close to the diastolic and systolic pressure. It's also due to the buildup of resistance. And then by the time we get to the capillaries, of course, it's relatively constant. The pressure is constant in those vessels. So this is what allows arteries to deal with the pulsation or the variable pressures that they experience. They have elastin in the tunica media, right? And it gives the arteries compliance, the ability to stretch, and recoil, the ability to push back against the blood, which actually does, I know it increases pressure on the blood, so it increases blood pressure, but it also helps the blood move forward. So in addition to there being a pressure gradient in arteries from high to low, there's also this push on the blood to keep it moving due to the recoil of arteries. And I think this next slide shows you a picture that sort of explains compliance versus recoil. So we have the heart, and the blood is moving into an artery with an elastic wall, and pretty high pressure, 120 millimeters of mercury, it's going to stretch that vessel, and luckily the wall is elastic, so it is compliant. It can take that. But then, of course, it recoils because those are elastic fibers that, when stretched, recoil. And so the recoil will push against the blood and help the blood move forward. It's pretty cool. Especially, we need that especially during ventricular diastole, so that the blood just doesn't stop moving when the heart's not pumping the blood out. Okay, now there's one disorder that's, I think, somewhat familiar to most people, and that's arterial sclerosis. It has an effect on compliance, and its layman's definition is stiffening of arteries. And the stiffening is due to is that the wall is thicker, either due to the buildup of plaque or there's been some sort of injury and scarring. There could be clots in there as well, but typically arteriosclerosis is talking about a thickened wall versus just a clot. And in these vessels, compliance and recoil are reduced. And the blood flow is going to be much more turbulent. It's not as laminar. Resistance is increased and the blood pressure is higher. The pressure in the vessel is higher. So the heart has to work harder to maintain the blood flow. Okay, let's look at blood flow in the capillaries. The velocity in the capillaries is slowest because these vessels have the smallest diameter, the most resistance. There's another reason though, and that is because there's this large cross-sectional area that all the blood gets to fit into. So this particular graph shows that the velocity of blood from the aorta to the capillary beds goes down. We knew that already. But then if we look at the total cross-sectional area, if you added up the space that can hold the same amount of blood that was in this artery, you would see that there's more space for the blood in the capillaries. So the blood slows down, slows velocity, okay? And the low pressure is advantageous because it prevents the rupture of delicate capillaries, right? We don't want the blood to burst a blood vessel. Okay, now we're going to look at blood flow in veins, and as we know, the blood flow, the velocity anyway, is going to increase, right? So why does it continue? Blood flow is maintained because there's still a pressure gradient. The lower pressure in the veins is due to the fact that they have larger lumens for better laminar flow, which decreases resistance. And the velocity speeds up as blood leaves these capillary beds, also due to vessels converging and having the same volume of blood fitting into less cross-sectional area. So it's got to speed up. So you can think of this picture that we had before. I go back. At the end of the capillary bed, if we just take this region right here, there's a large cross-sectional area that's holding a certain volume of blood. And if we add all three of these cross-sectional areas of little tiny venules, I guess, the volume of blood that can be held at any one time in the vena cava will be greater than what was in these venules. And so the blood gets pushed, and it gets pushed upwards towards the heart, in part due to the fact that our veins have valves, and the valves prevent backflow. In addition, we have a skeletal muscle pump, and I'm going to show you a picture of that, which relates to the valves. And then also our breathing helps promote blood flow, both in the arterial system and the venous system, but I think it's more interesting in the venous system. And there's always a little bit of venoconstriction. We call it venoconstriction because we're talking about constriction in the veins. This is exerted by the sympathetic nervous system. So let's first look at the valves and the skeletal muscle pump, which promote blood flow in the veins. You've learned already that the veins have valves. And if you have multiple veins entering this particular larger vein, you've got a large volume of blood getting pushed upwards. Well, just because it's being pushed doesn't necessarily mean it's going to go forward, but because the valves are present, anything that does go superior or upward isn't going to go backwards. It's not going to go backwards. And in addition, the skeletal muscles, as we move, push against our veins and likewise press on the blood and make it move superiorly as well. So they work in concert, those two. Now let's look at the respiratory or thoracic pump, which helps blood flow in general. Okay, when we inhale, what happens is our rib cage expands. So our thoracic cage, if you will, gets bigger. That means that there's a bigger volume. If there's a bigger volume, there's lower pressure. So there's low pressure. But the reason, in part, that our rib cage gets bigger when we breathe in is because our diaphragm presses downwards. So the diaphragm is a muscle between the thoracic cavity and the abdominal cavity. And here's, I just drew a line on the diaphragm, and it's depressed downward, making this rib cage bigger, and therefore the pressure has decreased. But we have increased abdominal pressure in that case. Okay? So what this does is, if we have high pressure here in the abdomen and lower pressure in the thoracic region, that promotes blood flow returning to the heart because blood flows from high to low pressure. Now let's consider when we exhale. Okay, so when we exhale, the diaphragm relaxes. And here we have a picture of the diaphragm moving upwards. It's relaxed. So what this does is cause the thoracic cavity, the ribs also kind of depressed, but there's muscles that control that. But you have a smaller volume in the thoracic cavity, so pressure is high. But when the diaphragm relaxes, that eases up on the abdominal pressure, so pressure's low in the abdomen. This is going to facilitate blood moving in the arterial system away from the heart. So when we exhale, that helps blood move away from the heart. When we inhale, that helps blood move back to the heart. This is called the respiratory or thoracic pump. Okay, now let's talk a little bit about blood flow and vasoconstriction. The term for vasoconstriction is vasoconstriction. And in general, when we think of constricting a blood vessel, like here in this diagram, it generally impedes blood flow, right? Because you have a narrower region. Generally, that's what we think of. However, if this constriction is not widespread and it's just local, like at this one spot, that could promote the movement of blood forwards and kind of giving it a push like elastic recoil in arteries. But this local constriction, vasoconstriction, helps in the movement of blood throughout the veins, which I've already mentioned, and it helps in elastic recoil, like it helps when the heart's in diastole and the arteries need to keep the blood flowing. So we'll see that sometimes constriction is helpful in moving the blood, and other times we think of it as impeding blood flow. So in the arterial system, elastic recoil promotes blood flow, and a little bit of vasodilation, just slight, will help lower resistance in arteries, so you'll get increased flow away from the heart. This is in the arterial system. But vasoconstriction beyond elastic recoil, when it's not just that amount of recoil, but further the vessel gets restricted even further, that decreases flow. In the venous system, we call it venoconstriction, I guess. Venoconstriction increases blood pressure, but because of these valves, and because we're going from high pressure to low pressure, it helps blood return to the heart. And so even though you think of constriction as increasing resistance, those vessels have so little resistance to begin with that just that little bit helps it return to the heart. That's the end of our first lecture. Thank you very much for listening.